AS Level Biology 9700, Paper 2, Variant 2, October November 2024, question number 5 and 6. And first of all, we will discuss question number 5. And question number 5 is malaria is an infectious disease caused by the Protoctis plasmodium. As part of its life cycle, plasmodium infects human red blood cells. Researchers can compare hemoglobin from the red blood cells of a healthy person with hemoglobin from a person with malaria. Throughout the world, most deaths from malaria are caused by P. vivax and falciparum. So, name one other species of Plasmodium that causes malaria. Students, there are many different species of Plasmodium which cause this malaria. So, Plasmodium falciparum is the most dangerous species, which is responsible for the majority of severe malaria cases and deaths worldwide. And the other P. vivax, as they have given its name, so this is also the most geographically widespread species. Then I have written the Plasmodium malariae and the Plasmodium ovale. So Plasmodium malariae species can be chronic, long-lasting infection. So this is very severe. It causes very severe type of infection in the body. And the other which I have written ovale. So this species is less common and primarily found in Africa. So it is now understood that there are two uh, subspecies of this ovale. So in this way, I would like to say we have to written only one name, but I have written these two names. You can write any one of these because I have given you an explanation about them, how they affect the human body. So in the laboratory, oxygen at different partial pressures can be bubbled through a solution of hemoglobin to determine the percentage saturation of hemoglobin at each partial pressure. A graph constructed from the results is known as an oxygen dissociation curve. So this figure 5.1 is an oxygen dissociation curve for normal adult hemoglobin in humans. So you can see a percentage on one side, there is given a percentage saturation of hemoglobin with oxygen and partial pressure of oxygen on x-axis and y-axis. They have given both of these, and you can see this uh, oxygen dissociation curve. So this is figure 5.1. Now question in the experiment used to obtain the results when uh, figure 5.1, the temperature and pH were standardized. So they have kept these standardized. Uh, so explain what the researchers would consider when deciding which temperature and pH to use in the experiment. Standardized. Standardized means to get good results without any ambiguity. So in this way, when the researchers decide, they keep in mind many different, you can say, thinkings. As for example, first of all, as we discuss about temperature. So the researchers would choose a temperature that will closely mimics the normal body temperature of the humans. As you know, the human has normal body temperature 37 degree centigrade or 37 degree centigrade or 98.6 degree Fahrenheit. 37 degree centigrade or 98 point. So, this is the normal body temperature, uh, which and this normal body temperature ensures that the hemoglobin behavior is studied under conditions that are relevant to how it functions in the body. So temperature significantly affects protein structure and function. You know, the highest temperature for protein, you can say the optimum temperature, which can be between 35 degrees centigrade to 40 degrees centigrade. So hemoglobin oxygen binding affinity changes with temperature. If temperature is more than normal, so the oxygen binding affinity will change. Using a physiological temperature allow for accurate representation of oxygen transport in the body. So researchers would select that temperature which will be very close to the body temperature. So same with the pH, and they will select the pH that will represent the normal body pH of the blood, that is 7.4, or you can say the neutral pH, because if there is change in the pH, it will influence the uh, affects the body. So it will also affect the hemoglobin molecule and its ability to bind oxygen. So deviations from this physiological pH can alter the shape and position of the oxygen dissociation curve. So for instance, a lower pH and would shift to curve to the right, indicating a reduced oxygen affinity. So standardizing temperature and pH ensures the experiment is reliable and reproducible by other researchers. So using these conditions also ensures that the results are biologically valid and applicable to understanding oxygen transport in the human body. And in simple terms, we can say that uh, both when we state that the physiological temperature and pH and explain why they are important, we can say that it is important for this standardization for reliability and validity of any of the experiment. So standardizing these conditions is essential for obtaining these reliable and comparable results that accurately reflects hemoglobin function in the human body. So I think they gave the results, accurate results, you can say. So I have explained the answer. Now you can see researchers would choose a temperature of approximately 37 degrees centigrade, which is the normal body temperature. So temperature significantly affects protein structure and function. The proteins are denatured at high temperature. So if temperature is high, more than 37 degrees C or 40 degrees centigrade, so it will affect the protein structure. So protein structure will be it will be denatured. So in this way, we can say the function will also affect the if there is change in the structure. So hemoglobin's oxygen binding affinity uh, changes with temperature. Second, standardization was kept in pH. So the researchers would select a pH that presents the normal pH of blood, that is 7.4, which is neutral. Deviation from pH. If there is change in the pH, can alter the shape and position of the oxygen dissociation curve. So in this way, I would like to say for the uh, reliable or accurate results. So I would like to write one more sentence over here. Standardizing these conditions is essential for obtaining reliable, valid, and comparable results. So these are the reasons. Now, next question is using a different, more rapid technique. Researchers compared the hemoglobin contained in red blood cell of a healthy person with the hemoglobin of a person with malaria who had been infected with P. vivax. So by analyzing the results, the researchers concluded that the oxygen dissociation curve of a person with malaria would be shifted to right. With reference to figure how a shift to the right of the oxygen dissociation curve would affect oxygen loading in the lungs. So, and unloading the respiring tissues in a person with malaria. So, the question is how loading and unloading will take place in the respiring tissues. And, and here I would like to explain first, then. So, here the question which we are going to discuss is the researchers would found the oxygen dissociation curve of someone with malaria is shifted to the right compared to a healthy person. So we need to explain the implications of this shift. So key concept in this case is the right shift of the oxygen dissociation curve. And the right shift of this oxygen dissociation curve means that hemoglobin has a lower affinity for oxygen. You can see in the figure 5.1 and the graph in which the oxygen dissociation curve has been shown. So this means more oxygen is released at a given partial pressure of oxygen. So it takes a higher partial pressure of oxygen to achieve the same level of hemoglobin saturation. So if we see that in the lungs, the partial pressure of oxygen is high, however, with the right shifted curve, hemoglobin in a person with malaria will have a slightly lower saturation at the same high partial pressure compared to a healthy person. Definitely in healthy person, the oxygen saturation will be high as comparison to a person with malaria. So this means that the blood is a person with malaria may not be fully saturated with oxygen as efficiently as in a healthy person because the red blood cells are damaged. The person who is suffering with malaria. So in respiring tissues, the partial pressure of oxygen is lower. With a right shift curve, hemoglobin in a person with malaria will release oxygen more rapidly, readily at the lower partial pressure. So we can say that this is happening. This is uh, several factors which are related to the malarial infection could contribute to the right shift. So I would like to say that in the lungs, this results in slightly less efficient oxygen loading, meaning the blood may not be fully saturated. And explain the other impact on oxygen is the in respiring tissues. In respiring tissues, first one is loading in the lungs, oxygen loading in the lungs, and unloading in respiring tissues. So, in respiring tissues, this leads to more efficient oxygen unloading means tissue receive more oxygen at the same partial pressure. So here I have explained the question. Now you can see the written one, which I have written for you. A right shift of the oxygen dissociation curve means that hemoglobin has a lower affinity for oxygen. You know, if it is this dissociation curve is shifted to right, it means the oxygen saturation has been decreased due to which it is shifted to the right. So this result is slightly less efficient oxygen loading, meaning the blood may not be fully saturated. It has the blood has less oxygen, so it is not fully saturated. And in respiring tissues, this leads to more efficient oxygen unloading. Definitely, less tissue, meaning tissues receive more oxygen at the same partial pressure. So the body's metabolic rate might increases during infection, leading to the higher carbon dioxide levels, which also shifts the curve to the right. And the person who is suffering with malaria and the person who is suffering with malaria has, you can say, less oxygen present in his body blood. So in this way, it will also shift on to the right. and a right shift in the oxygen dissociation indicates a lower affinity of oxygen for of hemoglobin for oxygen in the lungs. This results in slightly less efficient oxygen loading. Um, the blood may not be fully saturated. So in this way, I would like to say that it happens in the person. We can compare the person uh, with the who is suffering with malaria and the person who is a healthy person, and the oxygen uh, dissociation curve shift in this way onto the right side. Now the next question. A red blood cell that is infected with Plasmodium cannot carry out its function as effectively as a normal red blood cell. And you know, the red blood cells are damaged when the malarial parasites invade into the red blood cells. So the person become anemic. So when the person become anemic, you know, or oxygen saturation is also decreased in that person's body. So here describe how the size and structure of the red blood cell is related to its function other than the fact that it contains a very large number of hemoglobin molecules. So here I would like to describe this question in this way, that is uh, the question is on red blood cells. We will focus on the red blood cells, that is the red blood cells uh, are also called as the erythrocytes, and their structure is related with its function. So they are circular biconcave in nature. So they are circular biconcave nature. They are thick at the edges and they are thin in the center. They are non-nucleated. They don't have the nucleus. They lack a nucleus. We can say that, and it increases the surface area. You know, the shape of this red blood cell increases its surface area, and due to large surface area, it is able to carry more oxygen. So it contains hemoglobin, red color hemoglobin, which carries the oxygen, which act as a carrier of the oxygen. So in this way, the oxygen binds with this hemoglobin, and then in the form of oxyhemoglobin, the oxygen is, uh, you can say, distributed throughout the body. So it has very unique structure, biconcave shape, and as you know, the it lacks nucleus, and you can say that its size is very small, as for example 7 to 8 micrometers in diameter. So small size allow them to squeeze through the narrow capillaries, ensuring efficient oxygen. You can see through blood capillaries, these red blood cell can easily pass. So if, and they can also shape their in this way dumbbell shape. When there is blockage, they can pass very easily in by changing their shape. So they are very flexible. So they have very flexible. So due to lack of a nucleus, uh, more oxygen carrying capacity uh, is there, or oxygen carrying capacity is increased. So in this way, I would like to say the red blood cells of a red blood cells uh, are related to its function. So the red blood cells, after this, already you know this explanation. So the red blood cells are very small in size, about 7 to 8 micrometer in diameter. The small size allow them to squeeze through narrow capillaries, ensuring efficient oxygen delivery. Through the biconcave shape of the red blood cell increases the surface area to volume ratio. So this maximizes the diffusion of oxygen in and out of the cell, making gas exchange more efficient. Mature red blood cells lack a nucleus. This absence allows more space for hemoglobin molecules, maximizing the oxygen carrying capacity of the cell. So the red blood cells are flexible, which allow it to change the shape and pass through capillaries. So in this way, it was all about question number 5. Now I would like to move towards the question number 6 and the transport of water. Question number 6 is the transport of water from the soil solution to the xylem of roots occur by the apoplast and symplastic pathways. Uh, Mineral ions can be transported, dissolved, uh, dissolved in water. So here we have to focus on first of all, two types of pathways, which are apoplast and symplast pathways. So these two different pathways, uh, when there is movement, apoplast pathway when the water moves through the cell wall only without disturbing the cytoplasm. But symplastic pathway, when the water passes through the cytoplasm from one cell to the other through plasmodesmata. So through cytoplasm, the water moves. So here I would like to the describe the transport of water in from the soil solution to the endodermis, soil solution to the endodermis of roots by the apoplast pathway, and explain why the pathway cannot continue at the endodermis. So here uh, the question focus on the movement of the water from the soil into the uh, cortex and then into the endodermis. So here I would like to explain this. The movement of water uh, in apoplast pathway, the movement of the water and dissolved substance takes place through the cell walls and intercellular spaces of a plant. So this is essentially movement outside of the living cells. So this is the movement of water and dissolved substance through the cytoplasm of cells connected by plasmodesmata. It takes place in symplastic pathway when water moves uh, through the you can say cytoplasm. Now, the endodermis is a layer of cells which surrounding the vascular tissues, xylem and phloem in the roots. So it has a special feature called the Casparian strip. So Casparian strips. So Casparian strip is a band of so B embedded in the cell in the cell wall of endodermal cell. So here, okay. So Casparian strips, a band of Siberian embedded in the cell wall of endodermal cells. So these are the Vexi waterproof Siberian. What is Siberian? Siberian and Casparian strips is a Vexi waterproof substance. So the vascular tissues that transport water and mineral upward in a plant. So the xylem transport the water. So it will act as a vascular tissue. So during the transport, the water from the soil solution to the endodermis of roots by the apoplast pathway, and when water enters from the soil through the root hair cells, uh, and intercellular spaces of the cortex. So this movement is driven by a water potential gradient. You know, water potential gradient, that is water always move from higher water potential towards the lower water potential. So mineral ions are also move by dissolving into this water. So by the same pathway, that is the apoplast pathway. So the Casparian strips which are present in the endodermis act as a barrier and which is impermeable to water and ions. So the Casparian strip forces water and minerals moving in the apoplast pathway to enter the cytoplasm of the endodermal cells. So this allows the plant to selectively control which minerals enter the vascular tissue. So the endodermis act as a filter, preventing potentially harmful substance from reaching the xylem. So Casparian streak is like a roadblock that forces everything off the highway and into a checkpoint. So in this way, the Casparian strips. Uh, okay, next. The water from the soil enter after this explanation. The water of Casparian strips and the Casparian which uh, do not allow the water to move inside. So in this way, I would like to explain the question answer which I have written. So the water from the soil enters the root through root hair cells and moves through the cell walls and intercellular spaces of the cortex. So this movement is driven by a water potential gradient from an area of higher water potential to lower water potential. You know, the movement of water takes place in this way. The water is in higher water potential into the soil, then it enters through root hair cells, uh, soil to root hair cells, and then from root hair cells to cortex, cortex to endodermis. So in this way, water movement takes place. So this movement is driven by water potential. So the Casparian strip in the endodermis is a barrier of suberin. So suberin is a waxy waterproof substance which do not allow the water to move through. So the this allow the water to selectively control which minerals enter pathway to enter the cytoplasm of the endodermal cell. So at that time, the water moves through the cytoplasm instead of moving through the cell wall because of the presence of these Casparian strips having fibrin inside. So in this way, the water will move through the cytoplasm at that time. So this allows the plant to selectively control which minerals enter the vascular tissue. So the endodermis act as a filter, preventing potentially harmful substance from reaching the xylem. So it was, I hope you have understood the answer because I have also written with explanation. So researchers investigated the mechanism of transport used for the uptake of potassium ions into root epidermal cells at different concentration of potassium ions into the soil solution. Compare table 6.1 to provide information about the two different transport mechanisms that were identified by the researchers. Net movement of potassium and membrane protein needed, yes or not, ATP used, yes or no, name of transport mechanism. So here you can see first column net movement of potassium ions against the concentration gradient. So membrane protein needed, yes, it is needed. It is needed and it is also needed. So ATP used, yes. Active transport, and you know students in active transport or uptake of the different substance when it takes place against the concentration gradient from lower concentration towards the higher concentration. At that time ATP is required because they have to move against the concentration gradient or from lower concentration towards the high concentration. So the process which takes place or the mechanism which takes place is called as the active transport. Or and in this way, when they have to move in this way, they need energy. And the other is down the concentration gradient. And down the concentration gradient means they have to move from high concentration towards the lower concentration. So membrane protein is needed in that case, but ATP is not used because this is very passive movement. They have to move slowly, very slow movement takes place. So they don't need energy. So this type of name of transport mechanism is the facilitated diffusion, in which the substance move from their high concentration towards lower concentration or down the concentration gradient. So it was all about question number 156. In this way, we have completed paper 2. And please like, share, and subscribe my channel. Thanks for watching.